Good morning, everyone, and welcome. I'm Dr. S.K. Singh, and today I will be guiding you through an overview of the management of cultures used for microbiology analysis. In this presentation, we will explore the different techniques and importance of managing microbial cultures effectively. We'll cover key concepts, methods, and best practices to help you understand how cultures are used in microbiology. I'm glad to have you all here, so let's get started. Let's start with why culture management truly matters. Ensuring proper management of microbial cultures is essential because it directly impacts the accuracy, reproducibility, and reliability of microbiological testing. When cultures are handled correctly, the test results become trustworthy, which is critical for making informed decisions in our work. It also plays a key role in maintaining compliance with important regulatory and quality standards like ISO 17025, CLSI, and GLP. These standards help us ensure our processes meet industry expectations and legal requirements. Additionally, good culture management helps minimize risks such as contamination, misidentification of microorganisms, and biosafety hazards. This not only protects the integrity of our results, but also keeps our work environment safe. Now, let's clarify some key terms. Microbial culture refers to the growth of microorganisms under controlled conditions. Think of it as cultivating bacteria or fungi in a lab setting. Inoculum is the material we use to introduce microorganisms into the culture media, essentially the starting point for our cultures. And a reference strain is a well-characterized microorganism, like E. coli ATCC 25922, which we use as a standard to compare and validate our testing procedures. All these elements are interconnected, and understanding them helps us maintain high-quality, reliable microbiological practices that align with our overall goals. Today, I want to focus on our objective: standardizing and making our culture management traceable. This means implementing consistent practices that we can track and verify at every step. To achieve this, we rely on a solid regulatory framework and established standards that guide our processes. One key source is ISO/IEC 17025, which sets the requirements for laboratory competence. It ensures that our labs are capable of producing reliable and accurate results. Then, we also follow CLSI guidelines, which outline standard practices for handling cultures. This helps us maintain consistency and quality across all procedures. Additionally, good laboratory practice, or GLP, plays a vital role in ensuring our work adheres to ethical and scientific quality standards. We also prioritize biosafety standards, referencing manuals like the WHO Laboratory Biosafety Manual and the CDC NIH BMBL, to keep our staff and environment safe while working with potentially hazardous materials. All of these elements together form a comprehensive framework. They not only help us maintain high-quality, reliable culture management, but also ensure compliance with regulations. By integrating these standards into our daily practices, we reinforce the overall theme of quality, safety, and traceability in our work. When selecting and documenting bacterial strains, it's crucial to follow best practices to ensure consistency and traceability. Always use authenticated and traceable cultures, such as those from recognized sources like ATCC or NCTC, to guarantee the quality and identity of your strains. Proper documentation is key. Make sure to record the source of the strain, the date you received it, and the storage conditions to maintain its viability and integrity. Additionally, note the intended use for each strain, whether it's for susceptibility testing, identification, or other purposes. To keep things organized, assign a unique identifier to each strain, combining a lab-specific code with the date of receipt. For example, if you're working with Staphylococcus aureus for antibiotic susceptibility QC, you might label it as ATCC 25923. Following these steps helps ensure your strains are well managed and your results are reliable. Let's now turn our attention to labeling and traceability, which are essential for maintaining the integrity of our cultures. First, it's important to label all components clearly. This includes noting the organism name, the specific strain or ID, the date of preparation, and either the expiry date or passage number. These details help us track the history and viability of each culture. In addition to labeling, maintaining a thorough traceability record is crucial. This can be done through a culture logbook or a laboratory information management system, or LIMS, entry. These records should detail every step, ensuring we can trace the origin and handling of each culture. Also, preserving a chain of custody for distributed materials ensures accountability and proper tracking across different locations or users. Moving on to culture storage systems, there are primary storage types to consider. Short-term storage typically involves refrigeration or working stocks used for immediate experiments. For long-term preservation, cryopreservation in liquid nitrogen is the gold standard because it keeps cultures stable over extended periods. Speaking of stability, it's vital to avoid repeated freeze-thaw cycles, as they can damage cells and alter the culture's characteristics. To protect against this, we use cryoprotectants like glycerol at concentrations of 15 to 20%. These agents help safeguard cells during freezing and thawing, maintaining their viability and integrity. Overall, proper labeling, careful record-keeping, and effective storage strategies are key to ensuring the reliability and reproducibility of our work. These practices form the backbone of good laboratory management and help us uphold high standards in our research. Let's talk about the preparation of working cultures. First, we start by retrieving the cultures from the master stock, ensuring we do this under strict aseptic conditions to prevent contamination. Once we have the culture, we need to revive it and grow it until it reaches the target phase, typically the logarithmic or exponential growth phase, which is when the cells are most active. Before using these cultures, verifying their purity is crucial. For example, by measuring OD 600 to standardize the inoculum density, ensuring consistency across experiments. Now, moving on to the quality control of these cultures. We check for purity through methods like gram staining and subculturing to confirm that no unwanted microorganisms are present. To verify the identity of our cultures, we can perform biochemical tests or use advanced techniques like MALDI-TOF mass spectrometry. Counting viable cells through CFU enumeration helps us assess the health and concentration of the culture. Lastly, to ensure the genetic stability of our strains, especially for reference strains, we often use PCR fingerprinting, which confirms that our cultures haven't undergone unwanted genetic changes. All these steps are vital to maintain the integrity and reproducibility of our experiments, tying back into the overall theme of reliable microbiological practices. Let's talk about subculturing, also known as cell passaging. This is a crucial step in cell culture because, as cells grow and divide, they eventually need more space and fresh nutrients. So, subculturing involves transferring some of these cells to a new vessel with fresh medium. This process helps maintain healthy, proliferating cells and prevents overconfluence, which can lead to cell stress or death. It's important to do this carefully and at the right time, usually before the cells become too crowded. When passaging, we typically use enzymes like trypsin to detach the cells from the vessel surface, then dilute and reseed them at a lower density. This ensures they stay healthy and continue to grow optimally. Remember, consistent passaging is essential for experiments that require a steady supply of cells, and it helps maintain the cell's characteristics over time. So, understanding and mastering subculturing is key to successful cell culture work, tying directly into the overall theme of maintaining cell health and experimental reliability. Let's talk about contamination prevention. First, maintaining good aseptic techniques is crucial. This means always working carefully to prevent any unwanted microbes from contaminating our samples. Using a biosafety cabinet, specifically Class II, provides a controlled, sterile environment to work in, reducing the risk of contamination significantly. It's also important to use sterile media and disposables. Things like pipettes, tubes, and media should always be sterile before use. Regular disinfection of surfaces and equipment helps keep the workspace clean and minimizes microbial presence. Another key point is the segregation of cultures by risk group. For example, placing environmental isolates in separate incubators from clinical ones helps prevent cross-contamination and keeps our work safe and reliable. All these steps are interconnected and essential for ensuring the integrity of our work and safety of everyone involved. Contamination prevention isn't just a single task. It's an ongoing process that underpins the quality of our laboratory results. Let's talk about biosafety and risk assessment. First, it's crucial to determine the risk levels associated with the biological agents we're working with. This involves classifying them into risk groups 1 through 4, with risk group 1 being the least hazardous and risk group for the most dangerous. Based on this classification, we select the appropriate containment measures, which range from BSL-1 to BSL-3 or BSL-4 to ensure safety. Next, proper personal protective equipment, or PPE, is essential. This includes gloves, lab coats, and eye protection, things that create a barrier between us and the biological agents. But PPE alone isn't enough. Comprehensive training on its proper use is equally important. We also need to have clear protocols for emergency spill situations. Knowing exactly what steps to take can prevent accidents from escalating and protect everyone in the lab. All these measures connect back to our overall goal: maintaining a safe environment while conducting important research. Proper risk assessment and biosafety procedures are fundamental to protecting both personnel and the integrity of our work. Now, let's talk about waste handling and decontamination, which are crucial parts of maintaining a safe environment. First, it's important to understand the regulatory expectations. This means we need to follow the rules set by local and national authorities to ensure we're handling waste properly. Before disposing of any materials, always autoclave them. This step uses steam sterilization to kill any infectious agents, making the waste safe for disposal. When collecting waste, use clearly labeled biohazard bags so everyone can easily identify the contents and handle them accordingly. It's also essential to follow local biomedical waste rules, as regulations can vary depending on your location. These rules guide how waste should be stored, transported, and disposed of safely. Regarding decontamination practices, a common method is using a 10% bleach solution or an equivalent disinfectant. This helps to inactivate any remaining pathogens on surfaces or equipment. Remember, proper contact time is key. Monitor how long the disinfectant stays on the surface to ensure it's effective. By adhering to these practices, we not only protect ourselves and others, but also ensure compliance with safety standards. This is all part of the larger goal of maintaining a safe, clean, and responsible working environment. Let's take a look at some common issues and troubleshooting tips. When working through any system or process, it's normal to encounter challenges now and then. The key is knowing how to identify and resolve these issues efficiently. First, always start by clearly defining the problem, understanding what's happening and when it occurs. Next, check for obvious causes, like incorrect settings or connections. If the issue persists, consult the troubleshooting guides or FAQs, which often cover the most common problems. Remember, systematic troubleshooting helps you narrow down the root cause without unnecessary guesswork. Also, don't forget the importance of documenting any recurring issues, so you can look for patterns and prevent future problems. Connecting these troubleshooting steps to our overall theme, it's about maintaining smooth operations and minimizing downtime by being proactive and methodical. Staying calm and focused when issues arise is crucial for effective resolution. Let's now turn our attention to the crucial aspect of documentation. First, we have mandatory documents that form the backbone of our compliance and quality assurance processes. These include a comprehensive culture inventory, which helps us keep track of all the cultures we work with. Next are SOPs, or Standard Operating Procedures, specifically for culture handling. These SOPs ensure that everyone follows consistent, validated methods to maintain culture integrity and safety. Deviations and corrective actions are also vital. They allow us to identify when something doesn't go as planned and to implement measures to correct it, preventing recurrence. This is key to continuous improvement. Audit trails are another essential element. They provide a detailed record of all activities and changes, ensuring traceability and accountability in our processes. Now, with the increasing reliance on digital systems, tools like Laboratory Information Management Systems, or LIMS, play a major role. They help us track sample history and results efficiently, ensuring traceability throughout the entire process. Overall, these documentation components are interconnected. They support transparency, compliance, and quality, which are the foundation of our laboratory excellence and trustworthiness. All right, let's wrap things up by highlighting the key takeaways. Systematic culture management is really the cornerstone of success here. It's about ensuring quality, compliance, and safety go hand in hand throughout the process. Following standardized SOPs is crucial because they serve as our roadmap, ensuring consistency and accuracy at every step. And don't forget, maintaining traceability, quality control, and biosafety at all stages is essential to protect both our work and our results. These principles not only support high standards, but also help us stay compliant and safe in everything we do. So, by integrating these practices, we reinforce the overall integrity and reliability of our work. That's the key to achieving excellence in culture management. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional information you might need. Remember, effective communication and collaboration are key to success, so feel free to contact us anytime. And with that, I want to sincerely thank you all for your time and attention today. We appreciate your interest and engagement. Thank you.